Today, we're going to pull back the curtain on drug recalls. We're going to unpack the science of safety and take a close look at the microscopic impurities that can pose a hidden risk in the medicines we take.
You see those news alerts, you hear the brand names, but have you ever really stopped to wonder what's going on? Why would a medicine that's been on the market for years, that millions of people use, suddenly get yanked from the shelves?
Well, a lot of the time, it comes down to finding something that just wasn't supposed to be there. For the public, the alarm bells really started ringing back in 2018. The blood pressure drug valsartan was recalled because of an impurity called NDMA. Then it happened again in 2019 with Zantac, for the exact same reason. And again in 2021 with the smoking cessation drug Chantix. Now, these weren't just random one-off events. They were all connected by a specific type of impurity that created a huge challenge for the entire pharmaceutical industry.
Okay, so let's get into it. What is this hidden danger? At the very heart of these recalls is a specific class of chemical compounds, and they've become, a top priority for regulators all over the world.
They're called N-nitrosamine impurities. And to put it simply, these are just unwanted chemicals that can accidentally form while a drug is being made. The thing that makes them a nitrosamine is that very specific chemical structure you see right there, that nitroso-functional group.
So here it is. This is the basic blueprint. See that N-N double bond O? That is the signature, the telltale sign of a nitrosamine. And those R groups on either side? Well, they can be all sorts of different things, which is why we talk about a whole family of nitrosamines, not just one single bad actor.
So why all the fuss? What's the big deal? Well, the stakes are incredibly high. Nitrosamines are classified as probable human carcinogens. They're also mutagenic, which is a sciencey way of saying they can actually damage our DNA, even at incredibly low, almost undetectable levels. On top of that, studies have linked them to a whole host of really serious health problems, from liver disease to even potential impacts on brain health. So yeah, this is why regulators take even the tiniest trace amounts so seriously.
So if they're that dangerous, how in the world are they getting into our medicine in the first place? This is where the story gets really interesting. It's basically a high-stakes chemical detective mystery.
And it is so important to get this straight. This is not about someone maliciously adding a harmful ingredient. Not at all. These impurities are the unintended fallout from incredibly complex chemical reactions. They're just unwanted byproducts that form when the conditions are just right, or I guess just wrong.
So, the chemical detectives have a list of main suspects. First up, it can start right at the beginning, with the raw materials themselves. The manufacturing process is a huge one. You get certain chemicals and conditions together, and boom, you can inadvertently create nitrosamines. Sometimes, and this was the big issue with brandotidine, the drug molecule itself can break down over time and form the impurity. And get this, it can even be something as seemingly innocent as cross-contamination from shared equipment or a reaction with the drug's packaging.
Okay, so with all these potential culprits, how do manufacturers and regulators even begin to manage this microscopic risk? Well, it turns out they've developed a very systematic, very clear playbook.
It's a mandatory three-step process. First, companies have to do a deep dive, a thorough risk assessment, where they look at every single ingredient, every single step. If they find any potential risk, they have to move to step 2, highly sensitive testing to see if the impurity is actually there. And if the tests come back positive, step 3 kicks in, report it to the authorities, and most importantly, fix the problem.
Now, a really key tool in that first step, that risk assessment, is something called a purge factor. This is basically a score that predicts how good the manufacturing process is at cleaning itself out. You can think of it like a rating for a water filter, right? A really high purge factor means the process is super effective at removing a specific impurity before it ever reaches the final product.
And that idea is put into action using decision trees just like this one. Now, you don't need to follow every single line here, but the big idea is that companies calculate what's called a purge ratio. If that ratio is really high, way over there on the left in the green, the process is considered safe. But as the ratio drops, you can see how the rules get tougher, moving you from just needing some data on file to needing mandatory, hands-on testing in that red zone.
And if they do have to test, the results they get dictate exactly what happens next.
If they find an impurity, but it's at less than 10% of the daily safety limit, no routine testing is needed. If it's between 10 and 30%, they have to do periodic checks. But if it's above 30%, every single batch has to be tested. And of course, if it's over 100% of the limit, that's when you inform the authorities and you recall the product.
Finding these things requires technology that is just almost unbelievably sensitive. We're talking about incredibly advanced techniques, things like liquid chromatography mass spectroscopy, or LC-MS. These machines are needed to find a substance at the parts per billion level. To give you some perspective, that is like finding one single specific drop of ink in an entire Olympic-sized swimming pool. It's really a testament to how far this science has come.
So, what does all this mean for the future of the medicines that we all rely on every day? Well, it really points to a new era of vigilance and, frankly, collaboration.
This is not a problem that one group can just solve on its own. It absolutely requires a partnership. You've got the manufacturers on the front lines, and they're responsible for designing safe processes and managing their supply chains. And then you have the regulators, like the FDA, who set the safety standards, provide the guidance, and act as the ultimate public watchdog.
And I think the bottom line is this: making sure our medicine is safe isn't a one-and-done deal. It's not a destination you arrive at. As our science gets better, as our ability to detect these impurities improves, the standards and the processes have to improve right along with them. It is a continuous, never-ending cycle of learning, adapting, and improving.
Which, leaves us with one final, fascinating question to think about. In a world that's built on such complex, large-scale chemistry, where reactions can create countless unintended little byproducts, is the idea of 100% purity even possible? Or is the real future of drug safety all about managing that risk down to the lowest, safest level we can possibly achieve? Definitely something to think about.